Good afternoon, students, and welcome to the final practice problem week session. And I guess you guys are having your exams either today, tomorrow, or in upcoming short time. So be prepared, be well prepared, and today will be the last session. We'll solve some problems corresponding to lecture week 8. Let's wait for 5 more minutes, and I'll keep admitting students who are requesting to join this call. The same instructions as previous classes. If you had attended the previous sessions, please turn off your mic and video chat during this session. Do not fret, this will be an interactive session. So any interaction with me or via answering the questions will be through the comment box. You can type your opinions, your answers, and any communication with me through the comment box. And let's all keep our mics and video chat turned off for smooth conduction of this session. And we will start at 3:05. Let me wait for some more students to join so that yeah, both of the parties can benefit together. And all the best for your exams again. Please go through the lectures, and questions will be of the same pattern, MCQs. You just have to revise a bit. That's why these problem sessions, practice problem sessions, are here. Yeah. Yeah, it's already 3:05, and I see some students are asking about the question and the exam pattern. So, for your information, I would like to tell you that I am equally unaware of the maximum number of marks that are uh, going to be your final exam, but the questions will be much similar I think there will be MCQs, but I am not sure. See, it depends on the instructor. While I am just a TA and I am here to help you with the problems, so I literally do not know do not have that information. What kind of total mark or weightage will the questions carry? But there will be different sections of MCQs relating to all the corresponding lecture weeks. As long as you guys are thorough with your lectures and are attending the practice problem session, solving the problems, I do not think it will be a problem of how you get the questions in exam. So whatever questions you get in the exam, if your concepts and basics are clear, you can easily pass through them, right? So do not worry much about the pattern. And to be honest, even I am not aware of the pattern. So let's start on with the lecture lecture 8 practice problem session. Let me share my screen. Do not worry, and I do not think the exams will be too much difficult for you guys if you're following with the course uh, faithfully. Let me share my screen. And, yes. So, can you see my slides, students? And are they changing? If not, then please check your network connection. And if my voice is clearly audible, then we can start this session. Just quickly let me know in the chat box if you are okay with the slides and my voice, the audio. Great. So once again, I welcome you to NPTEL Nanotechnology in Agriculture practice assignment week 8. So this is the final set of lectures, and we will solve some questions related to lectures in the week 8. The instructor for this course is Professor Mainak Das from IIT Kanpur, and my name is Ashutosh Behra. I'm a PhD PMRF scholar from IIT Kanpur. So we follow the same pattern as we have been following all this while from practice week 1 to practice week 7 till now. So we'll answer a set of 10 multiple choice questions which have reference to the slides or to the lectures that you encounter in that particular week. So today we are going to do week 8 lectures, and I'll post the question. There will be 4 options, and I'll ask the students to think about what is the correct answer, and I encourage all the participants to answer. I see there are today is a thin population attending this class because I know some people are having exam. So just try to participate so that it will be beneficial for you as well as me. So let's go on with the first question. Do not shy out and be confident enough to answer. So the first question that on the screen, reducing the amount of salt from water is called dash. What is the process in which amount of salt in the water is removed or the amount is reduced? Option A, desanitation. Option B, desensitization. Option C, desalination. Or option D, none of the above. So you can see the question on the slide, and I'm reading it out for you again. Reducing the amount of salt from water is called dashed. I don't think it will be a great hurdle for you guys. It's a very easy question, and you might be just able to answer if you read the question thoroughly. And yeah, there are already some answers. Nidhi is saying desalination. Abhinav, Surbhi, Gaurav, desalination. Veer Chandra is saying desensitization. Okay. Any more answers? I think participation is too thin today. Hardly 10 participants. So let's not waste any further time and go through the options. So the correct answer to this question is C, desalination. So most of the students who have answered C, desalination. Very good. So removing or reducing the amount of salt from water. So salinity. Salinity is a measure of the amount of salt in any solution. So we say the seawater is saline because seawater has lots of dissolved salts like NaCl, KCl and other salts. Mostly, predominantly NaCl. So the source of salt for human beings is seawater mostly. And we evaporate in salt flats or salt ponds and then we create salt. So desalination is a technique which is now gaining uh, popular following because desalination is a process in which we can harvest the salinated seawater into and convert it into a manageable form of drinking water or irrigation water. We know that seawater is a highly salinated or highly salt containing water and it cannot be used for any industrial uses or irrigation purposes for agriculture. So we have invented such nanomaterials which can desalinate the seawater into pure water or not pure water into a form of water which can be used in irrigation, which can be used for drinking and many other purposes. So this is the process of desalination of seawater. What is desensitization? The other option that people were confusing about. Desensitization is a treatment technique used to modify how sensitive an area is to a particular stimuli. Desensitization means so a particular area or a particular part. For an example, to make it easier for you guys to understand, let's say your skin is sensitive to touch, pressure, pain, and heat, and such particular stimulus. So these are all different kinds of stimuli: touch, pain, pressure, heat. Desensitization is a technique in which regulated exposure to such amount of pain or touch, like with practice, the area of skin or the part which has receptors will get desensitized, means it will not respond to the certain levels which was earlier used and which could cause a response. So let's say a pin prick, a single pin prick or a single touch from a hot object is able to generate a stimulus or is able to put your hand away from the pinprick. So if you desensitize your hands or if you desensitize your hands over little bit of heat exposure daily, then you would not feel that much of pain that you used to feel earlier. So your hands are now desensitized to that heat. This technique is used to decrease or normalize body's response to particular sensation. So it's a biological technique in which our body's response to particular sensation is decreased. The example that I gave was heat sensation. So if you touch a hot object, And if you keep doing it regularly, practicably with low amounts of heat, then you might grow desensitized to that stimulus. So what exactly happens in desensitization? So this is a phenomenon of receptor desensitization, in which there is a receptor for an agonist or a molecule which has a receptor on the cell or body, and there is a therapeutic effect, whatever effect the agonist or something is trying to mediate. Now, down regulation of agonist receptor over time. What happens in the presence of agonist? These receptors are internalized into the cell membrane. This will bind the agonist molecule and will go inside the cell membrane. So with time, in the presence of agonist, the number of receptors on the cell surface will decrease. That's why now the same amount of agonist will give reduced therapeutic effect. So this is a condition called desensitization, and it is a phenomenon of receptor desensitization. One example of desensitization. So which was one of the options in our question. So this is the term desensitization. And desanitation, that we all know. Sanitation means maintaining hygiene and disinfecting the places of microorganisms or disease causing germs. So desanitation means we are removing sanitation. So that is also not the correct answer. And desalination, the topic of interest to us, as I told you earlier, desalination uses reverse osmosis technology to separate water molecules from seawater. So mostly this technique is used to desalinate seawater by a technology called reverse osmosis. Now, osmosis, what is reverse osmosis and how is it different from osmosis? What happens in osmosis is you have a semi-permeable membrane separating two different solutions, and the two different solutions have different solute and solvent concentration. Let's say you have highly concentrated salt solution on one side of the membrane and lesser concentrated salt solution on the other side of the membrane. Now what will happen? Through a semi-permeable membrane which allows certain substances to pass through and does not allow other substances. Let's say the semi-permeable membrane is based on the pore size of the filter. Now the filter will allow only small molecules like water molecules to pass through in the two solutions, but the solute molecules or the salt will uh, remain static and will not move across the membrane. Now what will happen? Water from its higher concentration to lower concentration will pass through the osmosis the semi-permeable membrane, and this process is called osmosis. So water in its high concentration is at the freshwater side, and water at low concentration is at the seawater side, because in a in a bucket of seawater there is more salt and less water than in a bucket of fresh water. So that is the technology of osmosis. Now what is reverse osmosis? Reverse osmosis causes the change in directionality of the flow of water. So now the water from seawater can flow back into the freshwater side when application of pressure. So that is the process of reverse osmosis. So desalination is using reverse osmosis technology to force the fresh water out of seawater and to leave behind the salts with the application of a semi-permeable membrane. Water from the ocean is forced through thousands of tightly wrapped semi-permeable membranes under very high pressure. So this is what I was explaining you through words. So there are semi-permeable membranes which upon application of pressure, water from the sea or seawater can be forced to produce fresh water. The membranes allow smaller water molecules to pass, leaving salt and other impurities behind. So as we learnt earlier, this is the process of osmosis. Water molecules are small enough to pass through the semi-permeable membrane, but larger molecules like salt and other impurities
cannot pass through the semi-permeable membrane, maintaining the concentration gradient. That's why we apply pressure in reverse osmosis technology. And this is a, diagrammatic representation of how reverse osmosis works. Here, this membrane here shows semi-permeable membrane, and on the left side is seawater or contaminated water, and on the right side we have fresh water, let's say salt water. Now here the concentration of water is high, here the concentration of water is low. Concentration of water molecules, because let's say there are 100 molecules of seawater or contaminated water and 100 molecules of pure water. So here we have 100 molecules of pure water, but here in contaminated water we have, let's say, 20 molecules of contaminants and 80 molecules of water. So the pure water will have higher concentration of water molecules in comparison to the contaminated water. So the osmosis direction will be higher concentration gradient to lower concentration gradient. So this is how normal osmosis is. Now what is reverse osmosis? We apply pressure from the contaminant water side, which will reverse the flow of directionality of water from higher concentration to lower concentration and cause the flow of water from contaminated side to pure side, from lower concentration of water to higher concentration. That's how salt water gets converted into fresh water with the help of the semi-permeable membrane and pressure applied reverse osmosis. Now this semi-permeable membrane is of interest to us in nanotechnology because many nanomaterials have been very good candidates to replace the semi-permeable membrane. The addition of carbon nanotubes into the filtration membranes can fasten the desalination process. Membranes with dense polymer-based films loaded with carbon-based nanotube pores can help with in-depth desalination while reducing the overall cost factor. So this is how nanomaterials like carbon nanotubes are used in semi-permeable water filter to in the desalination process. And this is an example of carbon nanotube laden membrane polymer based filter. So we have feed water here, sea or brackish water entering. There are carbon nanotubes integration for membrane modification. So this whole membrane has lots of carbon nanotubes like this membrane integrated body, like a pipe, whole pipe containing lots of small carbon nanotubes, and fresh water is pumped here out. This is the semi-permeable membrane consisting of carbon nanotubes, and here due to application of pressure, the water in the seawater is now getting converted into fresh water. So the answer to the option, as correctly answered by many students, would be desalination, which is the process of removal of or reducing the level of salt in seawater to convert it into fresh water. Is it clear to the students? We learned a lot about what is desensitization first, which was the wrong option. And also we learned about desalination, what is osmosis, the process of reverse osmosis, the importance of semi-permeable membranes, and how nanomaterials can be used in semi-permeable membranes in desalination process. Is it clear to the students? Just let me know in the chat box if you are able to understand. Great, great. Let's move on to the second question. The second question is before you. In photosynthesis, dashed and dash and water are converted to dashed and oxygen. So this is a question about photosynthesis, which is discussed at length in this lecture week series. So basically it's asking you about what happens in photosynthesis. In photosynthesis, dash and water are converted into dash and oxygen. So if you are aware about the starting materials and the end products of photosynthesis, you will be able to answer this question. Option A, carbon, carbohydrate. Option B, carbohydrate, carbon dioxide. Option C, carbon dioxide, carbon. Option D, carbon dioxide, carbohydrate. What do you think will be the correct answer, students? Let's take a minute and look at the question and collect some answers. And I encourage all of the students to answer. We have thin population today. Great, I see some answers here. Many people are really answering well. Vaishnavi, Nidhi, Avinav, Vishnupriya, Sudbi, Gaurav, Monica are all answering D. Dheeraj has also answered D. Carbon dioxide and carbohydrate, which is the correct answer. Well done, well done. So in photosynthesis, as you guys have all correctly answered, carbon dioxide and water are converted into carbohydrate and oxygen. So the starting materials are carbon dioxide and water. So this is the statement which environmentalists and all the other scientists say. Green plants fix carbon dioxide. Green plants help in stopping pollution or reducing pollution. How? The carbon dioxide emissions in metropolis and from vehicles, from firewood burning and everything, and from exhalation of living animals are all assimilated into energy, which is carbohydrate, by plants. So plants are very, very important. If plants are not there, then we are all dead within 10 years. So in photosynthesis, plants fix carbon dioxide. Mostly plants. There are also other organisms like cyanobacteria and green alga, which are not basically plants, but they can also fix carbon dioxide via the process of photosynthesis, convert it into carbohydrate and oxygen, and both of them are major requirements for all animal kingdoms and classes. Carbohydrate serves as energy source, food, and oxygen is required for breathing for all of us. Now, what is photosynthesis? Let's go by the definition. Photosynthesis is the process by which plants use sunlight, water, carbon dioxide to create oxygen and energy in the form of sugar. So, this is exactly what the question was asking us. So, plants use sunlight as a catalyst to trigger the reaction in which water and convert carbon dioxide, which are obtained very easily as reactants of this reaction, and the end products of this reaction are oxygen and energy in the form of sugar, which is carbohydrate. And this is the whole reaction here with a cute cartoon. So we have carbon dioxide here from the carbon dioxide emissions gas cloud, plus water, the plant in its leaf in the presence of sunlight gives us glucose, which is a carbohydrate, and oxygen. So again, repeating what we have learned: light from sunlight, chlorophyll is the pigment or the protein that is present in green plants, which make them look green, and it is an important factor which traps light energy to make food in the sense of glucose or carbohydrate. Oxygen, the end product of photosynthesis, given off into the air for respiration and breathing of many organisms. Carbon dioxide enters through the stomata of leaves. So there are certain structures in the leaves of plants called stomata. Stomata stomata are certain specialized structures which help in gaseous exchange of the plant. They are kind of the lungs and noses and respiratory system of the plant. Stomata is actually an opening formed by two bean-shaped guard cells, And these guard cells have lots of chloroplast harboring the chlorophyll pigment. So stomata, the guard cells of stomata are also the site for maximum photosynthesis in green plants. And here water is easily absorbed through the roots, as you guys all know, and starch, which is converted and stored as food in other parts of the plant. So starch, glucose, sucrose, and any kind of other sugars and carbohydrates are the end products of the photosynthesis reaction, which the plant requires for its own growth. And we as animals and other food chain survivors, we assimilate those carbohydrates into our own body for nutrition. And briefly, there are two stages of photosynthesis reaction. If you break down the photosynthesis reaction, there are two stages of photosynthesis. One stage of photosynthesis is called light reactions. Light reactions are all those reactions which happen in the presence of sunlight, or which require sunlight for the reaction to begin. And all light reaction happen in a structure called thylakoid, which is present inside chloroplast of a plant. So chloroplast is a double membraned organelle, like we have many organelles in an animal cell like Golgi apparatus, endoplasmic reticulum. We have mitochondria, one of the most important organelles, and we have nucleus and all these sorts of things. So plant cells have something called chloroplast, which harbor all these disc-shaped uh, coin-shaped uh, pro- coin-shaped structures called thylakoids, which are the center for light reactions. So all light reactions or light-dependent reactions happen in the thylakoid, and majorly the light reaction catalyzes oxidation of water molecule into oxygen. So removal of hydrogen water molecules converts the splitting of, this is also called splitting of water or photolysis, converts the water molecule into oxygen molecule. So number one is thylakoid, and there are other energy producing reactions in which ATP and NADPH are generated, which are the major source of energy currency. So all energy currency in the body is ATP and NADH. And there is a second set of reactions or second stages of photosynthesis in which all the reactions do not need the presence of light. Therefore, they are called dark reactions. So photosynthesis can be divided into light reactions and dark reactions. Light reactions are those dependent on sunlight to carry out the reaction, and dark reactions, unlike their name suggests, they do not occur in the dark. They occur side by side with light reactions in the day, but they do not require sunlight as a factor uh, for driving the reaction. And and it is this dark reaction in which the actual fixation of carbon dioxide into sugar molecules or into carbohydrate molecules take place. So in summary, photosynthesis takes up carbon dioxide and water molecules, converts it into oxygen and sugar molecules, which all of you guys have answered very correctly. I hope it is clear to the students the basics of photosynthesis and the kind of reactions, the starting products and the end products. I promise you there will be other upcoming questions about photosynthesis and chloroplasts, which will make us go deeper into that and help us understand it better. If yes, then please let me know in the chat box if it's clear to the students. Cool, cool, cool. Let's move on to the next question. The next question is also uh, about photosynthesis. In photosynthesis, water to oxygen conversion is called dash. Option A, light reaction. Option B, dark reaction. Option C, color reaction. Option D, none of the above. Let's take a minute and collect some answers before we move forward. What is the process of water to oxygen conversion called? Which phase of the photosynthesis catalyzes this reaction? Conversion of water molecules to oxygen. You can say oxidation of water molecules to oxygen. So just for your knowledge guys, a pro tip to remember what is oxidation and reduction. Oxidation is always addition of oxygen or removal of hydrogen. You can remember it like this. And a reduction reaction is addition of hydrogen or removal of oxygen. It's easy to understand this way. So the water is getting converted to oxygen, so hydrogen is removed from the water. That will be an oxidation reaction. Let's see if people have answered. Great, great. Vaishnavi is saying A. Nidhi is also saying A. Vishnu, Priya, should be Avinav, Gaurav, Rocky are all saying A. Yeah, yeah, yeah. I knew it would be an easy question for you to guys to answer. So the correct answer is light reaction, option A. As we discussed at length in the previous slide itself, the two phases of photosynthesis, light dependent reactions occur in thylakoid, converting water to oxygen. So option A, light reaction, is the correct answer. So let's learn a little bit about this conversion. So as we all have studied yet, water is an important raw material for photosynthesis because water is one of the starting materials, water and carbon dioxide, two important raw materials for photosynthesis. Now what happens during the light reaction and why is it called light reaction? During the light reaction, light energy is utilized, and by using this light energy, water is split to form oxygen and hydrogen, and electrons are released. So the water molecule is split using the energy from sunlight into hydrogen and oxygen molecules, and many electrons are released. So the purpose of light reaction is to catalyze this step and to generate electrons. Now these electrons will be used and will be channeled and will be flowed through the membrane of chloroplast, through the thylakoid membrane, and then help in generating energy via production of ATP and NADPH. So this process of water splitting to form oxygen and hydrogen in the form of light is very famously known as photolysis of water. You must have heard this term or read it in many places in books and in science articles. Photolysis, the term, if you break it down, photo means light. Since it's the form of light reaction, utilization of energy from light energy from sun. So it is photo. And lysis means breakdown. So breakdown of water molecule into its elemental oxygen and hydrogen using the energy of light is known as photolysis of water. And this is the first reaction to happen in photosynthesis, and it is a part of light reaction, photolysis of water. And this is the representation of photolysis of water. So H2O in presence of enzyme, this enzyme is a plant enzyme, gives protons 2H plus 2 electrons plus half molecule of oxygen. So 2 molecules of water will produce 1 molecule of oxygen, 4 molecules of H plus ions or protons plus 4 electrons. So these H plus ions will be further used for synthesizing NADP or for making NADP. So can you guys, I have a quick adjoining question. Can anybody from the students tell me what is the full form of NADP? You can type it in the chat box. What is the full form of NADP? What does NADP stands for? I hope you can hear me. Do you know guys what is NADP? You can just expand it. Can you just expand it for me? It's fine if you it's fine if you know, otherwise I'll let you know. These molecules. Great, we have correct answers. Vishnu, Priya, Nidhi are all giving very great answer. Congratulations. Great, great. Vaishnavi, you too. Nicotinamide, adenine, dinucleotide, phosphate. So all these students are very correct. NADP is an energy currency molecule, and it expands into nicotinamide, adenine, dinucleotide, phosphate. Nicotinamide, adenine, dinucleotide, phosphate. It's a complex benzene ring nucleated structure. And light reaction is the process of photosynthesis in which solar energy gets converted into chemical energy and is stored in the form of energy carrying molecules such as ATP and NADPH. So this is the summary of light reaction. Solar energy, which is light energy, converted into chemical energy in the forms of ATP and NADPH. Now let's talk about photosystem 2. What is photosystem 2? Photosystem 2 is a membrane protein supercomplex that executes the initial light reaction of photosynthesis. So this is a protein supercomplex or a protein complex association of many different proteins which carries out the light reaction in the chloroplast, in the thylakoid membrane of the chloroplast. When a pigment in PS2 absorbs light, it gets excited. The energy is then transferred to neighboring pigments multiple times through the process of resonance energy transfer. So this pigment which absorbs light and gets excited is the chlorophyll molecule. This is a very important characteristic of green plants. The green color that we see in plants is due to chlorophyll, which reflects green wavelength, which is perceived by our eyes. That's why plants appear green. So plants lacking chlorophyll will appear red or any other color but not green. So this chlorophyll pigment in PS2 absorbs light energy, gets excited, and this excite, energy excitement is passed on to neighboring pigments multiple times through a process phenomenon called resonance energy transfer. Now this is a phenomenon in which we have ground states and we have excited states of different molecules and different atoms. Now if the band gap between the ground state and the excited state is quite similar, then electrons can jump from one excited state to another excited state of a different molecule. So through that, there is resonance energy transfer between adjacent pigments. After the energy reaches reaction center, it is transferred to P680. So there is something known as a reaction center and the discharge center called P680 because it corresponds to 680 nanometer wavelength of light and it is present in photosystem 2. The electron is then transferred to an electron acceptor. The energy produced is used to split water and release oxygen. So this is the whole summary of the process of splitting of water or photolysis of water. So this is a complex diagram. You need not focus on everything. You can just follow my arrow. So here the first step is photon or light striking the photosystem 2 complex. So this green, purple and everything, this is the complex of photosystem 2 consisting of all these proteins. Now in photosystem 2, the chlorophyll will absorb the photon and then get excited and transfer it to the adjacent pigment via resonance energy transfer. Now this pigment will transfer it to the neighboring pigment, to the neighbor, to its neighbor, and it will reach finally the P680 reaction center. Now from this reaction center, it will go on to the electron acceptor, which is also a part of photosystem 2 complex. And there is also a water splitting enzyme complex, which will also use this energy of this electron transfer to split water into protons, H plus ions, and oxygen. So this is the whole process of splitting of water into oxygen, and it happens in the photosystem 2, which is also the place for occurrence of light reaction of photosynthesis. Is it clear to the students? We learned about the process of photolysis of water. Where do the protons and electrons generated end up? What is NADP? A little bit about photosystem 2 and how the process of water splitting is carried out. I hope it is clear to the students. Please let me know in the chat if it's clear, and we will move ahead. Yes, yes, got it. Great. Let's move on to the next question. Which of these is not a polymorph of TiO2, titanium oxide? Option A, anatase. Option B, azurite. Option C, brookite. Option D, rutile. Which of these is not a polymorph of titanium oxide? So titanium oxide is one of the great great nanomaterials that has a wide variety of uses, and many industries have applications for titanium oxide, and some other important nanomaterials would be cerium oxide, zinc oxide, and these oxide based metal nanoparticles are playing a major role in many industrial sectors and applicative processes where they're improving the yield and reducing the toxicity of whatever function they are performing. So titanium dioxide or oxide has been discussed at length in the week 8 lectures. So the question was, which of these is not a polymorph of titanium oxide, TiO2? Anatase, azurite, brookite, or rutile? Let's check some of these answers. Oh, students have nicely answered, very quick they are. I think they have revised very properly and went through their lectures. Great, great attitude. Nidhi, Vaishnavi, Avinav, Vishnu, Surbhi, Gaurav are all saying B. Azurite. Azurite is not a polymorph of TiO2. Let's see. You guys are all correct. Option B, azurite, is the correct answer. Azurite is not a polymorph of titanium oxide, while all other three, anatase, brookite, rutile, are polymorphs of TiO2. Now let us learn a bit about all these. What are polymorphs and what is the polymorphism? Polymorphism is the ability of a substance to crystallize into different crystalline forms. So basically, these anatase, brookite, rutile, these are all chemically titanium dioxide only. They are not much different. It's the same chemical and elemental composition. But what's different between all these, and why are these polymorphs? These are called polymorphs because the same titanium dioxide can crystallize into different forms, and these different crystalline forms have different kinds of properties. So these different crystalline forms are called polymorphs or crystalline modifications. Polymorphs have the same liquid or gaseous state, but they behave differently in the solid state. See, that makes sense, right? Because so if you heat a polymorph, or you try to bring the crystalline state into a gaseous state or a liquid state, then since the chemical composition and the elemental makeup is same, it's the titanium dioxide, everything will be a liquid or a gas of titanium dioxide. The only thing that is differentiating them is the physical form of crystalline form, which is different when they are in solid state. So polymorphs have same liquid or gaseous state, but they behave differently in solid state. These are very important properties, characteristic distinct from others when they are in solid state. There are four common polymorphs of titanium oxide, including anatase, rutile, bruchite, and TiO2b. So three of these were in the option: anatase, rutile, bruchite, and TiO2b. Usually, nano-sized anatase is easy to obtain, especially with a diameter less than 14 nanometers, while rutile presenting in bulk are considered as thermodynamically stable form. So anatase can be nano-sized, diameter less than even 14 nanometers, while rutile is in bulk. Rutile presenting in bulk are considered to be thermodynamically stable form. So naturally occurring TiO2 in nanoform is called anatase, crystalline nanoform, and in bulk form is called rutile. And here is a mineral picture of a mineral containing anatase. So this is a anatase mineral, or the TiO2 mineral. Looks so shiny in the grainy bedrock. And this is a lattice structure which is showing the arrangement of TiO2 lattices in rutile, anatase, and bruchite, which tells us the basic differences between them. So all these different lattices, so the purple atom that we see here is the titanium atom, and then we have oxygen atoms in coordination with that, and these form a tetrahedral, sorry, octahedral structure, and they are arranged. If you see, anatase is arranged in, they have different angles and arranged in a longitudinal cuboidal form. Breadthwise cuboid is rutile, and bruchite is in a cubic form. So these are different polymorphs of titanium oxide, and the arrangement of lattice structure of titanium oxide particles constitute the different physical form of the mineral. So rutile has different properties than anatase. As we saw the example, anatase occurs in nanoform naturally, but rutile occurs in bulk form. So the properties are different. While on the other hand, option B, azurite, that you guys have answered very correctly, is not one of the polymorphs of TiO2. Then what is azurite? Azurite is a soft, deep blue copper mineral. So it's not titanium mineral, it's a copper mineral produced by weathering of copper ore deposits. It is not one of the TiO2 polymorphs. So azurite, azure is always associated with copper. Azure means you must have heard of the color azure blue. So azure blue is the color of copper mineral, copper sulfate, bluish color, very beautiful color. And here is a crystal of azurite, which shows the azurite mineral. So this is copper oxide and copper sulfates and lot other forms of oxidized and reduced forms of copper sulfide, and which makes up azurite. So it's a copper mineral rather than TiO2, titanium mineral, while rutile, anatase, and brookite are TiO2 polymorphs. Now, is it clear to the students? You guys have answered very fast and very correctly. I'm happy to see that. Uh, but we'll go through all these questions, understanding the concepts behind them. So I'm just reconfirming. Is it clear to the students? Great, great, great job. Now, slowly, let's move on to the next question. Question number 5. Which of these properties is or are exhibited by titanium oxide? So it's again a question about in continuation with the properties of titanium oxide. Now, which of these properties, mind you, these are all applicative properties of titanium oxide. So, which of these properties do you think titanium oxide is good at? Is titanium oxide used for photoremediation? Is it used for water splitting, anti-fogging, or all of the above? So, which of these properties can be exhibited by titanium oxide? Let's take a minute to think and answer accordingly. Is titanium oxide being used in photoremediation? What is photoremediation? And is it used in water splitting or anti-fogging or all of the above? Okay, I see there are many answers already. The moment I read out the questions and students are ready with the answers, believe me, it could make any instructor very happy. So Nidhi, Vaishnavi, Vishnupriya, Surbi, Gaurav are answering D. So all of the above. Titanium oxide is used in photoremediation, water splitting, anti-fogging, is it? Let's check out what is the correct answer. And the correct answer is option D, all of the above. So all the students are very correct. Surprisingly, they are answering very nicely today. So titanium oxide has wide variety of uses in photoremediation, in water splitting, as well as anti-fogging. And let us check out one by one. What is photoremediation? Sorry, it's not photoremediation, it's phytoremediation. Sorry for the typo. There is nothing such as photoremediation. Phytoremediation basically refers to use of plants and associated soil microbes to reduce the concentration of toxic effects of contaminants in the environment. So phytoremediation. Phyto means plant life. Remediation. So usage by using plants, we have devised a way of remedying or restoring the quality and quantity of the environment. So that is in a whole process called phytoremediation. So basically, using plants and soil microbes to reduce the concentration of toxic effects of contaminants in the environment. One major function of plant as a phytoremediation technique is assimilation of carbon dioxide from CO2 emissions and turning it into carbohydrates and sugar using by the process of photosynthesis. So that is the most important phytoremediation that people are ignoring a lot and taking it for granted. But there are many other forms of phytoremediation, such as here is a study showing effectiveness of titanium dioxide nanoparticles in phytoremediation of antimony contaminated soil. So there is a soil which is contaminated. There is a patch of soil, maybe near to industrial area or nuclear power plant, which is contaminated with heavy metals like antimony and lead, silver contamination, mercury contamination. So all these contamination are very fatal. There are many diseases associated with heavy metal toxicity like arsenic and antimony and mercury. So mercury contamination causes a rare disorder called Minamata disease, which is prevalent in Japan because of nuclear radiation in Japan. So many kids and adults developed who were born during that time and later had incorporated radiations and mutations in their bodies, which led to deformities in their hands, arms, and skeletal structures. So such soils or crops or environment should needs to be detoxified so that our crops and the environment that we live in remains pure. So these plants, what do they do? They take up these heavy metals and other chemicals that are treated in the soil and assimilate them for their own use, so that the amount of heavy metals now or the toxicity of the heavy metals is now reduced, and the soil or the environment or the water or the treatment phytoremediated area is now usable for human. So we are basically very selfishly applying plants and nanoparticles to reduce the toxicity created by humans. So in this graph we see application of, so in the y-axis we see bioconcentration and translocation factors of antimony. So this soil was contaminated with antimony. The translocation factor and bioconcentration. So a plant is translocating antimony from the soil and accumulating in itself, freeing the soil from antimony. TiO2 nanoparticle concentration in milligrams per kilogram. So 1000 milligram per kilogram TiO2 nanoparticles. See, there is a reduction, steep reduction in bioconcentration and translocation factors of antimony. So it is reducing the bioconcentration of antimony in the soil at higher concentration of TiO2 nanoparticles. So TiO2 nanoparticles have found a usage in the phytoremediation technique. Now the other technique that TiO2 is used very frequently is in photocatalysis of water or photolysis of water due to its redox properties. And now how does it carry out the photolysis of water? So this here represents the energy band gap or conduction band and valence band of TiO2. Whenever energy is applied in the form of light, electron will jump from this lower valence band to the upper conduction band. Now this electron then can be used. Uh, Now this electron then can be used with atmospheric oxygen, because TiO2 nanoparticles also provide high surface area to generate superoxide radicals, and similarly the same electron can be used for production of oxygen from H2O with the process of radical formation. So OH radicals and O2 minus radicals, TiO2 can thus help in photocatalysis of water. And this diagram here also shows that TiO2 can act as an antimicrobial agent via the production of superoxide radicals and free radicals, which can attack the microbes or damage the membranes of many bacteria. Fungus can be used as antifungal agent, can be used to catalyze the splitting of water in the presence of light due to the excitation of electron from valence band to conduction band. Among the other semiconductors, TiO2 has become a promising material that has been widely used in photocatalytic process due to its superior properties. And what are these superior properties? Excellent optical and electronic properties, high photocatalytic activity, which we just saw here, high chemical stability, low cost, non-toxicity, and environmental friendliness. It is very environmental friendly, provides high surface area, has a perfect band gap length which can be excited by light, high photocatalytic activity, and high optical and electronic properties. So it makes TiO2 a good candidate for photocatalysis reactions. Now TiO2 is also used in nanocomposites, most importantly white pigment in the coating industry. So TiO2 is also used in the coating industry. Now what do we coat things with TiO2 in? It is widely used because it efficiently scatters visible light, thereby imparting whiteness, brightness, and opacity when incorporated into a coating. So this property of TiO2, which we also saw here, excellent optical and electronic properties. So due to its optical properties, it can impart whiteness, brightness, and opacity when it is applied as a coating because it efficiently scatters visible light. Now, where do we need this? In anti-fogging mirrors, which was our third option. TiO2 is used in anti-fogging mirrors as a coating because it can scatter light so easily. So it does not allow formation of fog or haze in the mirror, and it is easier for drivers to see through the mirror and windshields. So in windshields there is coating of TiO2, and in mirrors here is a diagram showing anti-fogging effect of automobile side view mirror. This is the normal conventional mirror and TiO2 coated mirror. So you can see the difference crystal clear due to TiO2 coating, which can effectively scatter visible light and impart whiteness and brightness and opacity. So TiO2 is also used in nanocomposites for coating different windshields and glass and mirror glasses. And it is used as photocatalysis agent. It is used as a uh, phytoremediation agent. So all of the above is the correct answer. TiO2 has variety of uses in phytoremediation, in photocatalysis of water, and in anti-fogging mirrors. Is it clear to the students? I hope you are able to understand. Yes, great, great, great. Excuse me. Let's move on to the next question. I hope you guys can all see the question. Gratzel cell refers to dashed. So this is the question about Gratzel cell. What does a Gratzel cell refers to? Option A, voltaic cell. Option B, Daniel cell. Option C, human cell. Or option D, dye sensitized solar cell. I'm sorry, I'm having a little cough today. Please excuse me. Let's focus on the question. Gratzel cell refers to a voltaic cell. Is it a Daniel cell, human cell, or a dye sensitized solar cell? Let's take a minute and consider some answers. My god, there are already so many answers. Nidhi, Vaishnavi, Gaurav, Roshan, Vishnupriya, Surbhi. Everybody is answering D. Dye sensitized solar cell. You guys are very thorough with all these lectures, seems like. You are well prepared for the exam. Great, great, because this is the correct answer. Gradual cell is also known as dye-sensitized solar cell. And you guys are all correct. Great job. Now let's understand what is a dye-sensitized solar cell and where does it get its name from, gradual cell. A dye-sensitized solar cell is the third generation of solar cells. It belongs to the thin film solar cell category. So this is the third generation of solar cells and it is a type of thin film solar cell. So we have most most big buildings now have relied on solar power to generate electricity, and as you know, solar power is more cleaner and greener, environment friendly form of generating electricity than hydrothermal power, which was and which also is currently the major source of electricity production. So almost 50% of the power comes through hydrothermal ways and thermal power plants where we burn coal to generate electricity via steam engines, and some other cleaner energies such as wind turbines, solar cell, and tidal energy turbines, and etc. So the current generation and the current uh, revolution demands for more eco-friendly and higher output generating uh, higher energy output generating methods, and thin film solar cell category is one of them. So dye sensitized solar cell is a thin film solar cell and is one of the third generation of solar cells. This advanced solar cell transforms visible light into electrical energy. The dye within the solar cell generates electricity while in contact with sunlight. So if you draw similarities between this solar cell and photosynthesis, in photosynthesis, light energy got converted into chemical energy, while in a solar cell, light energy is converted into electrical energy. And how? With the help of the dye and conductor system, which we'll see shortly. The dye in contact with the conductor system will generate electricity. These solar cells are among the cheapest solar cells available on the market. They were invented by Dr. Brian Regan and Professor Michael Grezel in 1991. So it's not a very way old innovation. It's only 20 to 25 years back that this solar cell method was discovered, and it was invented by Brian Regan and Professor Michael Grezel. So where from where it derives its name, the Grezel cell, and it is one of the cheapest solar cells available in the market. So despite being eco-friendly and it is also very cheap, so that many common people and other all classes of people can afford it, and it can be widely used and it can be popularized. So here is a representation of a dye sensitized solar cell or a Grezel cell. Sorry, yeah. Now if we follow the mechanism in which this works, so here is the source of our light energy, the sun, which is always shining and is the source of all energy for organisms on earth. So the light energy here comes, and this is the structure of solar cell. So the first layer the light energy encounters or the light encounters is conducting glass or FTO. So conducting glass will allow the light energy to collect it and pass it through. And the immediate layer here is made up of titanium dioxide, which is an integral part of dye sensitized solar cell. So this titanium dioxide forms a semiconductor kind of layer here. And the following layer, the titanium dioxide layer, is sandwiched between conducting glass, and the dye layer. So here, conjugated to the titanium dioxide is a dye, and this dye absorbs the solar energy or the light energy, and the electron from the ground state of the dye excites into the excited state of the dye or the chemical. And this band gap, this ground state to excited state band gap, is quite similar to what the valence band to conduction band gap is with TiO2. Especially the conduction band here is at par level with the excited state of the dye. So what can happen now via resonance energy transfer? This electron, which has now excited from the ground state of the dye with the help of absorbing the sun light or light energy, this electron has now excited into the excited state of the dye. This electron can jump via resonance energy transfer into the conduction band of titanium dioxide, and this process is called electron injection. So the excited state of the dye will inject an electron into the semiconductor titanium oxide packed nanomaterial conduction band. Now there is a wire connecting which will transfer this electron from the conduction band here. So all this material is TiO2. So this TiO2 electron from this conduction band of TiO2, now that it has entered the titanium oxide molecule, this electron will flow through the wire, get conducted. This is showing the ampere and voltage here. The electron will now flow through the wire, and whatever load is connected here will receive electricity. Now what happens when the electron finishes its job? There is something called a counter electrode on the other side of the solar cell, and the space between the counter electrode and dye is filled with electrolyte, generally consisting of iodide ions. Now the electron from the counter electrode will release, and there will be a redox reaction which will now regenerate the electron, giving it energy back for the excited state electrons to come back to ground state. And this cycle will again continue when light energy will again be absorbed by ground state electrons of the dye. Electron will get excited to the excited state. There will be electron injection due to resonance energy transfer, similarity of levels of conduction band of TiO2 and the dye. Then the electron will flow through the wire, work its load via electricity, come to the counter electrode where it reduces the electrolyte again, and there is a redox reaction going on. Now the electrolyte getting reduced will produce the maximum voltage and bring back the electron from excited state to the ground state, and the process can again start over. And there are many benefits of using a dye sensitized solar cell. These cells are commonly used in portable gadgets and low density applications. So the electrons generated via this do not have very high output or maximum output. So that is one of the disadvantages of dye sensitized solar cell, that the output generated can only power uh, general daily to daily light gadgets and all. But the kind of electricity that we generate from hydrothermal and thermal coal power plants are way much higher and can run millions of buildings and apparatus and lights. But what are the advantages of a dye sensitized solar cell? Here are some of the advantages. Since it has a strategic structure, it can extract more protons from the sun rays. So the sun rays containing proton, due to its structure and arrangement of TiO2 and the dye sensitized solar cell can extract more protons from the sun rays. It has a low production cost. Cheap materials are used to manufacture this solar cell. So the major factor that makes this an attractive candidate is because it is very cost effective, and many classes of people can afford using this form of cleaner and greener energy. It does not require any apparatus and is more economical than semiconductor cells. Does not require any specific sophisticated apparatus. This generation of solar cells is capable of absorbing fluorescent light and diffuse sunlight. Not only sunlight, it can even absorb diffuse sunlight and fluorescent wavelengths also. It can work effectively in cloudy climatic conditions. So little amount of sunlight can also generate electricity in cloudy climatic conditions when where there is no constant input of sunlight. Still, the dye sensitized solar cell can work in presence of little or basal amount of sunlight on a cloudy day. It has a good shelf life, which makes it a much more likable product. And this is a picture representation showing a dye sensitized solar cell. And these grids here represent the titanium dioxide grading material with dye conjugated to it. The gaps here are filled with electrolyte, and there is a counter electrode which will replenish the electron by reducing the electrolyte and replenishing the dye. I hope students were able to understand the working of a dye sensitized solar cell and the advantages of using it. Is it clear to the students? You guys are answering very awesome, and all your answers are correct. We're just trying to learn a little bit about the understanding and background of all these answers and questions. Great, great. Let's move on to the next question. Which of these are not part of the chloroplast? So it's a question about photosynthesis again, and now the organelle chloroplast. Which of these are not part of the chloroplast? Option A, crista. Option B, manganese cluster. Option C, chlorophyll. Option D, thylakoid membrane. What do you think will be the correct answer? Which of these are not a part of chloroplast? I guess it's very easy if you play elimination. If you can rule out the options which are a part of chloroplast, then you can easily find out which is not a part of chloroplast, or the other way around. Let's take a minute and collect some answers. What I was expecting, and I know that people have already answered. And yes, see, four people have already answered. Vaishnavi, Gaurav, Vishnupriya, Surbhi are all saying A. Let's wait for a minute and collect some more answers. Nidhi is saying B. Okay. Students who haven't gotten a chance yet to answer, please come forward and type your answer. It's not of a serious competition anyway. You're free to answer, and it will be a very productive session if you come forward and try answering these questions. It's okay if your answers are not correct. We will eventually see what is the correct answer, and we spend so much time on understanding why the correct answer is the correct answer, right? So, students who have not yet gotten a chance, please try to answer. We are reaching towards the end of these sessions because this is the seventh question, and we have three more questions left till we reach the last 10th question. Yeah, let's not uh, let's not delay it any further. And all of you guys are very correct. Answer to this question is Krista, option A. People were confusing. Nidhi was confusing with manganese cluster, but it's fine. Manganese cluster is a part of chloroplast. We will all see all these functionalities in the chloroplast, and we'll also learn about Krista. What is Krista? So let's learn about the chloroplast. This is how a chloroplast cross section would look like. So the chloroplast is a double membrane structure. Now there are not many double membrane structures in cells. If you look at them, the cell itself is a single membrane structure. Not all cells. Animal cells have lipid bilayer. Plant cells have cell walls in them. So double membrane structures are organellic structures present inside the cytoplasm, and they have their own machinery and harbor special structures and protein performing different functions, such as the chloroplast here that we are seeing. The mitochondria is also a double membrane structure. Endoplasmic reticulum, Golgi apparatus are other organelles but which are not double membrane. So the chloroplast here has an outer membrane and an inner membrane, and the space between the outer and inner membrane is called intermembrane space. And inside of the inner membrane, we have a ground substance or an aqueous liquid called stroma, and coin-shaped structures called thylakoid. So each one of the coin-shaped structure is called thylakoid, and one such stack of thylakoids is called granum, stack of thylakoids. And there is something called lamella which is connecting these different stack of thylakoids or granum, and all these building kind of structure connecting with pipelines of lamella, stroma lamella are present on the ground substance of stroma. And inside this thylakoid is the lumen of the thylakoid, where we will see many important reactions take part. So the thylakoid are membranous structure. The chloroplast is in itself is a double membranous structure, and on the stroma ground substance there are stacks of granums. Singular unit are known as thylakoids, and they are connected via stroma lamella or lamella. And inside the thylakoids there is the lumen, which is the center for many important reactions that the chloroplast carried out, such as photosynthesis. And this is a representation of photosystem. So we learned we saw the three options were manganese cluster, and what were the other options? Chlorophyll and thylakoid membrane. So clearly thylakoid membrane is present in the chloroplast. And this is a representation of photosystem 2. So this is a thylakoid membrane, stroma, the outside of the thylakoid lumen. So the outer area inside of the thylakoid, and this is the thylakoid membrane. And this protein complex here is called photosystem 2. So there are pigment molecules, chlorophyll molecules. Chlorophyll A special pair, which will transfer the electron into the primary electron acceptor. This is also known as P680, the 680 nanometer wavelength responding to the protein. And this is a reaction center, which is the primary electron acceptor. And this energy, electron transfer energy from the light, will be now used to split the water with the help of water splitting enzyme. And this is also called light harvesting complex, the complex harboring all these pigment molecules, which will act via resonance energy transfer, harboring the light energy or passing the excited electron into adjacent pigment molecule, and finally sending it to the reaction center, and through that to the primary electron acceptor. So photosystem is also a part of thylakoid and hence chloroplast. And now what is a manganese cluster? A tetramanganese cluster, Mn4, four molecules of manganese are clustered together, resides at the active site of photosystem 2 in green plants, and in certain bacteria and alga. It catalyzes the light-driven water oxidation reaction to generate dioxygen. So the enzymatic complex, which utilizes the electron transfer energy in the photosystem 2, from the light energy. There is an enzyme complex which utilizes this energy and splits the water into oxygen and hydrogen. That enzyme contains a tetramanganese cluster, which is commonly known as manganese cluster. So that enzyme is also a part of photosystem 2 complex. And this photosystem 2 complex proteins is as a whole present on the thylakoid membrane. Thus, it is also a part of chloroplast. The question that was asked was, which of these is not a part of chloroplast? And the option were crista, manganese cluster, and chlorophyll and thylakoid. So chlorophyll is the pigment molecules. We see here there is chlorophyll here, which is also a part of reaction center. We transfer the electron. The photosystem 2 is a part of the thylakoid membrane. Thylakoid membrane is a part of chloroplast. So all these three options would not be correct. So the correct answer is crista. And manganese cluster is a cluster of four manganese atoms present in the enzyme which catalyzes the splitting of water with the help of sunlight. Now, what is a crista? This is a shoe-shaped structure of mitochondrion, a single mitochondrion, showing its double membranous structure, the outer mitochondrial membrane, inner mitochondrial membrane, and the space in between them called inner membrane space. And here, similarly, the space inside the inner mitochondrial membrane is called matrix, and there are ribosomes in mitochondria, and mitochondria has its own uh, DNA which replicates on its own independent of the parent DNA. There are granules, starch granules, food storage, and mitochondria is commonly the place where ATP synthesis occurs, or the energy for the cell act for the activities that are going on in the cell comes from the mitochondria, ATP generation. So here is a diagram showing the presence of ATP synthase particles which will synthesize ATP via the electron transport system that generates the electron, inner membrane space matrix, and the inner membranous lining is folded into finger-like projections called cristae. And there is mitochondrial DNA, inner membrane, and outer membrane. So a crista is a fold in the inner membrane of a mitochondrion, which provides larger surface area, accumulation of greater mitochondrial protein, ATP synthesis, accommodation of electron transport system, which are integral part of generating the energy currency of the molecule. It gives the inner membrane its characteristic wrinkled shape, providing an amount of large surface area for chemical reactions to occur on. So this is what I was talking about. The finger-like infoldings of the inner mitochondrial membrane, known as crista, provides large surface area for chemical reactions to take on. That is the reason why crista are formed finger-like in the mitochondrial membrane. So the answer to this question, as you guys have all answered correctly, is crista. Crista is part of a mitochondrion and not chloroplast, whereas manganese cluster and chlorophyll and manganese cluster, chlorophyll and others, thylakoid membrane are part of chloroplast. I hope it's clear to the students. If yes, then please let me know in the chat box. Great, great, great. Let's move on to the next question. Now here is a calculation question, and it's about the energy equation. If you guys are familiar with the energy equation, you can solve this very easily. If the wavelength of light wave is doubled, the resulting energy is dashed. So the question is saying, if we double the wavelength of light, what will happen to its energy? Will the energy be doubled? Will it be four times? Will it be halved? or will it be 1/4 of the previous energy? So we have original wavelength. Now we are using a wavelength which is double the original wavelength. What will happen to the energy of the wave or the particle? Come on, students. If you remember well the energy equation, you can easily calculate this. What will be the resulting energy if the wavelength of the light wave is doubled? Let's wait for a minute and collect some answers. Vaishnavi has wrote A and C. I don't think it can be doubled or halved at the same time, right? Only C. Okay, okay, okay. Thanks for clarifying. Nidhi is saying C. Vishnu Priya halved. Rashmi is saying doubled. Komal halved. Vaishnavi is saying C. Let's see what is the correct answer. The correct answer is option C, halved. It cannot be doubled. Very good. Great guys. You guys are understanding what the question is demanding. If the wavelength of light is doubled, then the energy will be halved. And let's see how it is. So here is a small calculation of the question. So if the photon energy or the wave energy is E, it can be represented by this famous equation called Max Planck's energy equation involving the Planck's constant. H here is the Planck's constant. E is equal to h nu, and nu here is the frequency. Nu can be written by as c by lambda, and lambda here is the wavelength of the photon, and c here means speed of light, 3 into 10 power 8 meter per second. So photon energy E can be written as E is equal to h nu or E is equal to hc by lambda. Now from that relation, E is inversely proportional to lambda. And a simple calculation, given lambda 1 is lambda and lambda 2 is now 2 lambda. This is what the question was. If the wavelength is doubled from the original and E1 is equal to E, then E1 by E2 is equal to lambda 2 by lambda 1. E by E2 is equal to 2 lambda by lambda. E2 is equal to E by 2. So it's as simple as this. So if you add a 2 lambda here, then the E will be E by 2, because energy and wavelength are always in an inverse relation with each other. Remember this, guys. Energy is inversely proportional to the wavelength. So whatever question follows, you can solve accordingly. Now, if somebody asks you if the wavelength is 1/3 of the energy, let me ask you this question very quickly. If the wavelength is 1/3 of the energy, 1/3 of its original, then what will be the answer? Instead of doubling the wavelength, let's do it 1/3 of the wavelength. Now, what will be the new energy? Can you quickly answer this question? If the new wavelength is 1/3 of the original wavelength, how will the energy change? Good, Nidhi. You are very correct. 3 times. Tripled. Rabisha. Very good. See, it's as simple as that, because the, the simple relation is energy is always inversely related to wavelength. So that's it. The correct answer is energy will decrease by 2 times. Vishnu Priya, very good. Correct. 3 times. Let's move on to similar question. Now the question is, if the frequency of a light wave increases to 3 times of original, what will be the resulting energy? It's a slightly twisted question. So we were dealing with wavelength last time. This is frequency this time. What will be the energy if frequency of a light wave increases to 3 times of original? Very good, very good. I can see like people are really answering very fast. 3 times, or A, option A will be the correct answer. Undoubtedly, undoubtedly. Very good. And it will follow the same equation. So the Planck relation, E is equal to h nu. So you always remember this equation, very important equation. Energy is equal to h nu, and the also the other important equation is the kinetic energy equation. E is equal to half mv square, right? You remember these two equations and you can solve any such problems. So Planck relation, E is equal to h nu. Energy is always directly proportional to the frequency, and energy is always inversely proportional to lambda, or the wavelength. And h and c are here constants. E is energy, h is the Planck constant, 6.6 into 10 power minus 36. Nu is here frequency, this is nu. C, speed of light, 3 into 10 power 8 meter per second. And lambda is the wavelength of the wave, or the photon, or whatever entity we're calculating. So owing to this Planck relation, E is equal to h nu. So we have 3 times, we have increased the frequency 3 times. So the energy will increase 3 times, which is the correct answer. Energy will increase by 3 times as well, because of the direct relation of energy uh, and the frequency. Now let's move on to the last question of this session, of the entire uh, NPTEL Nanotechnology TA session. The last question: the ultraviolet light has dashed frequency and dashed wavelength than visible light. So it's a comparative relative answer. In comparison to visible light, what is the frequency of ultraviolet light and what is the wavelength of ultraviolet light? Option A, higher frequency, higher wavelength. Option B, higher frequency, lower wavelength. Option C, lower frequency, higher wavelength. Option D, lower frequency and lower wavelength. Now, what do you think is the correct answer? In comparison to visible light, guys, ultraviolet light. If you remember the electromagnetic spectrum, which consists of all kinds of waves, then you can answer this question. This is pretty easy question, and I have just hinted you the biggest help you can ever need in this question. It's okay if you do not, if you answer it wrongly, it's fine. We will see, and I'll make sure that you batter it inside your concepts, the concept of frequency, wavelength, and any other question that we were solving during all these problem solving sessions. I make it sure that you guys understand it very well, so that in future, whenever you face such questions or you face a problem like this, you are ready with the answer. So let's see some answers. A, okay. Roshan is saying A. Rashmi, higher, lower, B. Gaurav is saying B. Komal is saying C. And oh, Surbi is very clever. She is answering both B and C. My God, that won't be the that wouldn't be the right approach. Nidhi is answering B, and Vaishnavi is saying D. Roshan corrected himself. B. Okay, I think you guys need a good basics to understand this. So I'll explain it. Let's see what is the correct answer. Option B is the correct answer, guys. So all those who have answered correctly, the right answer is option B. Congratulations. Now here is a representation of the whole range of electromagnetic spectrum, their applications, and the range of wavelength and the range of frequencies. So let me walk you through this. The first kind of waves that we encounter, radio waves, and this is a chart for wavelength, 10 to the power 3 meters. So do you see how high is the wavelength for radio waves? So this is visible light, where we have zero point 0.5 into 10 to the power minus 6 meters, or in micrometers. In comparison to the visible light, radio waves have far greater wavelength, 10 to the power 3 meters. Then comes microwave, 10 to the power minus 2. Then comes infrared. So all the infrared, microwave, radio waves come before visible light in the wavelength spectra. So all these have greater wavelengths than visible light. Then comes the ultraviolet, which was the question of our interest. Ultraviolet rays, X-rays, gamma rays. So as we move in this direction from radio waves to gamma rays, there is a decrease in wavelength. That means radio waves are very high wavelength and gamma rays are the least wavelength. Similarly, if you see the frequency scale, 10 to the power 4 is the scale frequency for radio waves, microwave 10 to the power 8, infrared 10 to the power 12, visible rays 10 to the power 15, ultraviolet 10 to the power 16, 18, 20, it increases till gamma rays. So what do you observe is frequency is following the opposite trend. Frequency is increasing from radio waves to gamma rays, while wavelength was decreasing from radio waves to gamma rays. So it is as evident as simple, because wavelength and frequency are inversely related to each other. If you follow the same primal equation of energy, E is equal to h nu and nu is equal to c by lambda. Now nu and lambda are again inversely related. So when a wavelength of light will increase in one direction, in this direction, the decrease wavelength of light will decrease in this direction, while frequency will increase in this direction. So if you point out two arrows, gamma rays to radio waves, wavelength will increase and frequency will increase in this direction from radio to gamma waves. I hope it's clear to the students, and here are some of the application: approximate scale wavelength. What are the wavelength scale in comparison to the radio waves? Buildings, humans, butterflies. So these are the size ranges in which wavelengths of these waves fall under. And always you remember, there is an inverse relationship between frequency and wavelength. Both cannot increase in one direction. So for an ultraviolet light in comparison to visible spectrum, ultraviolet will have higher frequency and lower wavelength. So that is the correct answer, option B, higher frequency and a lower wavelength. Is it clear to the students? This is the most basic concept you all of you guys should be clear with, because many big engineers and scientists, they have all these basics rooted in them, that frequency and wavelength are inversely related and one cannot increase, both cannot increase in the same direction. An ultraviolet has a higher frequency than the visible light and a lower wavelength than the visible light. I hope it's clear to the students. Yes, great. With this, I'll stop sharing my screen, and I see there are some queries in the chat box. Let me first stop.
presenting. Yeah. So what is the paper pattern? We'll have options for all the questions or for some of the questions only. I think the paper pattern will be the same kind that you have been receiving, the assignments and the type of questions that we were solving, multiple choice answers. But I'm not sure because I'm a TA for this course and I'm not the one deciding the question paper. But I think the professor and the NPTEL team will go on with the same format, though I cannot surely assure you. But you guys need not fear because if you're prepared well, then uh, you can handle any kinds of question. I think it will be MCQs as per my prediction. So do not worry much. Just focus on your preparation. So please upload video on YouTube. Yes, yes, I will definitely because this session is getting recorded and all the videos will be uploaded on YouTube. It will take some time because these are 2 hour long sessions. And see, you guys told me that you had an exam on Sunday, so I had to request the NPTEL team to prepone this session on Saturday. But then I came to know that some people are also having exam on Saturdays, so I didn't knew then when to conduct this session, and I couldn't do anything about it because it takes times and efforts to create these slides. I saw a comment on YouTube yesterday asking, "Sir, could you please upload these presentation?" But the fact is that I was still making these presentations; they were not yet ready. How can we see earlier problem-solving sessions? So, Nidhi, all these problem-solving sessions recordings are available on YouTube, and even the presentation slides that I have been presenting, all these problem-solving sessions will be available in Google Drive, and the link for the there is a Excel sheet which contains the link for all the YouTube videos and all the Google Drive link for folders, and that is already mailed to you guys. You just need to check your older mails. There will be a mail from NPTEL Nanotechnology Agriculture giving you the link for this Excel sheet, which will contain all the 8 recordings for the YouTube video. The eighth one I'm going to upload today, and all the slides for the problems that we have solved during this course of time. So again, for your convenience and for all the students who are attending this class right now, let me copy paste the link for both the drive and the YouTube link, right? Let me do it here, then you do not have to go back to the mail. And but it's a loss for those who didn't attend today's session. Let me just quickly save your time. Search my YouTube. So I'll paste the link to the playlist which contains all the videos, and today's video will be uploaded after 5 p.m. when we end this session. And so that playlist contains starting from practice problem week 1 to practice problem week 8, and you can visit them anytime. Those are all 2 hour recording lectures which were live sessions that we took. You can just or you can just go to the YouTube, search my name Ashutosh Behra for nanotechnology and agriculture, and you'll get it. I think this yeah, I've copied the link. This is the link to the YouTube. This is the link to the YouTube uh, playlist, which contains all the 7 to 8 recordings. And do not worry, I'll upload this recording very soon as I end this session, and you can access this. Just wait for a minute. Let me access my drive and share the link for you guys to view all these slides that I have been preparing for you guys. Or you can just see this. These all these have been provided to you, but I am providing it to you separately because I want all of you to excel in your exams. This is the link for the Google Drive folder, which contains all the slides from starting from week 1 to week 8. I've uploaded all the slides with their correct answers and explanation. So even if you go through them, that will be a quick revision for you guys. But once again, I will encourage all of you to go through your lectures. If you have some pending lectures left, just go through them, skim through them. It's not very difficult to follow. And anyways, the question, we have already dealt with some of the questions, important questions in these problem solving sessions. And I don't think it will be a problem for you guys. Is there any other query that I'm missing on? Because there are, while I'm presenting, I'm not able to check the comments so soon. I have to wait for a minute and log in through my phone or from other device. Nidhi was asking about earlier problems solving sessions. Rasmi sir, please upload video on YouTube. Yes, I'll upload it. Do not worry. The session is still here. Previous year question paper available? No, I cannot. I cannot help you with that because I have no knowledge about this. As you guys know, these schemes of this new scheme of NPTEL teaching, in which PMRFs like us have been asked to take problem solving sessions. These are very unique. I might not be your TA next time you take this course, or next time somebody is following this course. Might be or might not be. So I do not have any access to the previous year question paper. I guess you can find it on the internet, or somebody on Quora, you can find it. But I'm sorry, I cannot help you with that. Let me just quickly skim through the. Rashmi is asking, sir, please revise all questions again. Oh my god. I already, I'm sorry, Rashmi, but we have already spent 2 hours. I can understand you had college today, but what I can suggest you is there is always this recording available. You can go to the YouTube channel or the link, watch any kind of recording, any week problem solving recording session that you have missed, or you want to follow it again, you can do that. But I'm sorry, I cannot discuss it again. Is there anything else that I'm missing out? Previous year question paper. Oh, there are. We'll have calculations too. Not much, I think. The kind of things that we had, not very petty calculations. If you have followed the lectures nicely, I don't think there will be much. And oh, there are lovely feedbacks on the comment sections too. Vishnu Priya, thank you, Vishnu Priya. Thank you, Rashmi. Yeah, all the best for your exams. Yes, Gaurav, making topics easy is what teachers like teachers should do. Even though I would say I'm just a student, so I'm a PhD student, as you guys all know. I've been putting it up on my intro slides. So I'm a PhD student at IIT Kanpur, and we are just doing our duty, PMRF TA duty, to teach people. So I'm not teaching you guys, but helping you solve these problems, making just slides and all for you. So thank you all. Yeah, I have shared the YouTube and the drive link again. I'll copy paste it again for some people are joining here again. So this is the drive link, guys, and the YouTube channel link. You can just go type it in the YouTube, Ashutosh Behera, my name, or you can just type the course name, Nanotechnology in Agriculture, and the videos will pop up. There are 7 videos currently, and I'll upload this last video. And I've already uploaded the slides for uh, the lecture week 8. Yeah, thank you for smooth conduction of this session, because I had given particular instructions that mics and video chat would be turned off. And thank you for bearing with me for these long 2 hours. And with this, I would like to end the TA session for this course. So we have together solved 8 weeks of lecture problems related to those lectures for nanotechnology and agriculture. And hope to see you soon. Bye. Take care and good luck for your exams. Thank you. I'll end this call for everyone.